What is photosynthesis? Most of you probably already know. Plants absorb sunlight to make food. It's what we were taught when we were young. But how does it work? How do plants convert light energy into usable chemical energy? To understand photosynthesis, we must look inside the leaf. Inside the leaf, there is an organelle called the chloroplast, a double-membrane organelle that has special structures called the thylakoid and the stroma. Photosynthesis also consists of two steps. There is the light reactions, which occur in the thylakoid membrane, and have photosystems, which create energy for the second step, called the Calvin cycle. The Calvin cycle takes carbon dioxide and converts it into usable chemical energy. Photosystems have chlorophyll molecules, a reaction center, and a primary electron acceptor. There are two photosystems, photosystem 1 and photosystem 2. In photosystem 2, light is absorbed by chlorophyll molecules and is transferred between chlorophylls to a special chlorophyll molecule. Simultaneously, electrons from the splitting of water are transferred to an acceptor in the reaction center, where the light absorbed by the special chlorophyll molecule excites the electrons to a new energy level, where it is accepted by the primary acceptor, and is transported through the electron transport chain through the cytochrome complex to the second photosystem. When the electrons are transported between photosystems, the energy that it gives off is enough to stimulate ATP synthase, create ATP, the energy molecule that powers several biological functions. In photosystem 1, the same thing happens. Light is absorbed and is used to excite electrons to be accepted by an acceptor. However, this time, the electrons come from photosystem 2, and these electrons are then used to reduce NADP+ to NADPH, an energy molecule that fuels the Calvin cycle, using an enzyme called NADP+ reductase. Both the ATP molecule and the NADPH molecule are transferred to the stroma for further use. In the other step, the Calvin cycle, carbon dioxide is converted to sugar using the energy made from the light reactions. There are three steps of the Calvin cycle: carbon fixation, reduction, and regeneration. In carbon fixation, carbon dioxide is combined with a 5-carbon acceptor using rubisco, a catalytic enzyme, and is then split into two 3-carbon acceptors. In reduction, these 3-carbon acceptors accept electrons from NADPH and are reduced in the process. 6 ATP molecules are also used in reduction to make the 3-carbon acceptors into a sugar called G3P, which is later turned into sucrose, a usable sugar. Then in regeneration, the remainder of the G3P molecules are used to regenerate the 5-carbon acceptor by using 3 more ATP molecules. Finally, the used up energy molecules get sent back to the light reactions to be recharged, and sent back to the Calvin cycle to power another cycle, to create more sugar so the plant can thrive. This showed a simple model that can create sugar. However, the Calvin cycle is repeated multiple times just to get one molecule of sugar.